After studying this module, you shall be able to know the role of metal ions and other inorganic elements in biological systems, classify the protein metallobiomolecules as electron carriers, as storage and structural moieties, and oxygen binding molecules. Identify the ways by which metallobiomolecules act in our body as transport and storage protein. Learn about the various protein enzyme metallobiomolecules. Classify these enzymes in the categories of hydrolysis, oxidoreductases, isomerases, and synthesis. Introduction. Metal ion plays an important role in biological system, either as metallobiomolecules or sometimes it exists as such. Detailed mechanisms, functions and importance of these metal ions will be discussed in subsequent modules. However, a very brief introduction about these metal ions is presented here. Non-protein metallobiomolecules. Photoredox. Radiant solar energy enters biosphere using magnesium porphyrin complex, chlorophyll. The process is also referred as photosynthesis, and almost all life depends upon chlorophyll and photosynthesis. The oxygen in the atmosphere is a byproduct of photosynthesis, and the foodstuffs are either parts of plants or animal which feed on plants. This is the structure of chlorophyll. Magnesium also plays an important role in muscle contraction and nerve irritability. Non-protein metal transport and structural metallobiomolecules. The first one, the bacterial iron transport. The agents responsible for iron transport into and within bacteria are known as siderophores. They are special chelating agents and are produced by bacteria and ejected into their environment together and transfer iron through the cell wall into the cell. The main problem which is encountered in iron transport is that it is not readily available to aerobic organisms in aqueous environment because of very low solubility product of ferric hydroxide at pH 7. Simple diffusion into cell could never fulfill the requirement of iron inside the cell. Siderophores form chelates with iron and transport the iron into the cell. In some cases, it has been seen that the siderophore releases the iron at the cell wall and iron passes through the alone, whereas in some other cases, entire complex coordination compound enters the cell and releases iron when required. Second one, skeletal. Calcium is present in the body in large amount than any other mineral element. About 99% of the body calcium is in the skeleton, whereas it is present as deposits of calcium phosphate in a soft fibrous matrix. The major inorganic constituent of bone is comprised of a crystalline form of calcium phosphate. Silicon appear to be an integral component of acid mucopolysaccharides and may have structural role in connective tissue, cartilage, skin and bone. Silicon is an essential element for growth and skeletal development in chick and rat. It is used in the form of silicon dioxide for structural purposes in diatoms, some protozoa, some sponges and one family of plants. Synthetic compounds such as silicons are also becoming very important and are finding many applications in medical science, as these can be used as artificial skin, etc. Phosphorus is found in every cell of the body, but most of it, about 80% of the total, is combined in the bones and teeth. About 10% of it is in the combination with proteins, lipids and carbohydrates and in other compounds in blood and muscles. The remaining 10% is widely distributed in various chemical compounds. The great importance of the phosphate ester in energy transfer is also known. Protein transport and storage protein metallobiomolecules. The first one, electron carriers. Cytochromes. Cytochromes as shown in this figure are heme proteins that act as electron carrier and are involved in photosynthesis. It consists of a porphyrin ring chelated to iron atom. Cytochromes are found in all aerobic form of life. They are also present in chloroplast for photosynthesis, but also in mitochondria and take part in the reverse process of respiration. It is important to note that iron functions well in the cytochromes because of the ferrous-ferric redox couple EMF and has a value in the appropriate range for life processes. Iron sulfur protein. Several non-heme iron sulfur proteins are involved in electron transfer. They are made up of peptide chains having cysteine amino acid moiety, where cysteine sulfur is coordinated to iron or cluster of iron sulfur atoms. The term redoxin is used for these having one iron atom, while those containing iron sulfur clusters are known as ferredoxin. Ferredoxin is the first electron acceptor in photosynthesis. Different types of iron sulfur protein are shown in this figure. Blue copper. It also acts as an electron carrier in photosynthesis and mainly occur in algae, green leaves and other plants. The different forms of blue copper are azurin, plastocyanin, stelacyanin and eumocyanin. Metal storage and structural metallobiomolecules. Ferritin. The regulation of iron absorption is largely carried out by the intestinal epithelial cells. The excess of iron can be stored with cell in non-toxic form by binding to protein called apoferritin in the mucosal cells. Iron 2, once within the mucosal cell, is oxidized to iron 3 and then after combining with apoferritin form iron protein called ferritin. Therefore, we can say iron can be stored as ferritin and can be released to usable form when required. This is the structure of iron in ferritin. Transferrin. Nearly all the iron released from the mucosal cell enters the portal blood, mostly in the iron 2 state. In the plasma, where pH is 7.4, iron 2 is oxidized to iron 3 and then picked up by the iron binding proteins transferrin. This protein is a glycoprotein of molecular weight 77,000 containing two similar but not identical sites that bind two atoms of iron 2 per molecule of protein to form a red ferric protein complex. Ceruloplasmin. It is a blue copper binding plasma protein which exerts as catalytic activity serum ferridoxic in plasma to convert iron 2 to iron 3 and therefore this metalloenzyme help in iron incorporation into transferrin. Oxygen binding metallobiomolecules, hemoglobin and myoglobin. The function of oxygen transport and storage in higher animals is provided by iron porphyrin complexes, hemoglobin and myoglobin. The former transport the oxygen from the lungs to site of its use inside the muscle cell and therefore the oxygen is transferred to myoglobin for use in respiration. The iron in hemoglobin and the myoglobin is in +2 oxidation state. Oxygen molecules coordinates to iron 2 from one end of heme and globin protects the iron 2 to be oxidized to iron 3+ by creating a hydrophobic environment so that the water molecule could not reach the site. Assuming there is no globin present, the oxidation reaction would have occurred and the product hematin in which iron is in +3 oxidation state must form. Hematin cannot bind oxygen, thus become useless for oxygen transport. Hemerthyrin. It is a non-heme oxygen binding iron protein using great variety of marine worms and phyla of marine invertebrates. It also contains iron 2 which binds oxygen reversibly and when oxidized to iron 3 metameritrin, it shows no tendency to bind oxygen. Two forms are known, octameric form and monomeric form. Each oxygen binding site contains two iron atoms and reaction take place via redox reaction to form iron 3+ and peroxide in both monomeric and octameric forms. Hemocyanin. Hemocyanin contains copper and bind one molecule of oxygen for every pair of copper ion. When oxygen is bound to hemocyanin, its color is blue. Otherwise, unbound hemocyanin is colorless. They are found only in mollusks and arthropods, Crustacea and arachnids. They occur dissolved in blood or hemolymph and usually comprise of nearly 95% of the total protein present. Each subunit contains two atoms of copper and polypeptide chain surrounding them. Molecular weight 50,000 to 74,000. These subunits further form aggregate with a molecular weight up to 9 × 10⁶. Vacuoles in the blood cells, vanadidocytes of the acetidine contains a vanadium protein complex dissolved in acidic medium. It has been established that carefully prepared vanadidocytes uptake oxygen reversibly. The next one, protein enzyme metallobiomolecules. There are many different ways whereby enzymes accelerate the rate of reactions, but here only those mechanisms will be discussed where metal ion plays some important role. Metal ion forms perform catalytic and structural role in proteins as discussed earlier. Enzyme form metalloenzymes, ternary enzyme metal substrate complexes in different ways, thereby catalyzing different reactions. Metal ions may also play important role in each of the 4 different mechanisms by which enzymes are known to accelerate the rate of chemical reactions. The first one, general acid-base catalysis. Second, covalent catalysis. Third one, approximation of reactants. And the fourth, induction of strain in the enzyme or substrate. Metal ions have ability to accept electrons and therefore these act as electrophiles. From filled orbitals may also act as electron donor, though to a limited extent. Thereby sometimes may also act as a nucleophile. Metal ions also supported to be excellent Lewis acid. Because of all these chemical properties, all metal ions have tendency either to form a sigma bond or multiple bonds. In addition, metals can serve as a 3-dimensional template for the orientation and binding of basic groups present on the enzymes or substrate. Metal ions accepting or donating electrons can activate either electrophiles or nucleophiles and therefore can act as an acid-base catalyst. Example include kinases, lyases, pyruvate decarboxylase where metal activates an electrophile, carbonic anhydrase where metal activates the nucleophile, pyruvate decarboxylase, carboxypeptidase, alcohol dehydrogenase where metal withdraws pi electrons, non-heme iron protein where metal donates pi electrons. Sometimes the coordination sphere of a metal may bring together enzyme and substrate or form chelate producing distortion in other enzyme or substrate. Example, in phosphotransferase, D-xylose isomerase and hemoprotein, metal ion produce strain effect to accelerate the rate of reactions. Stereochemical control can be achieved by presence of metal ions in enzymes such as pyruvate kinase, pyruvate carboxylase, adenylate kinase where metal ions gather and orient ligand. This is achieved by ability of the metal coordination sphere to act as a 3-dimensional template to hold reactive groups in a specific orientation. Enzymes can be further classified into many types, but also only those where metal ions play will be dealt here. Hydrolysis. These are the enzymes catalyzing hydrolysis of ester, ether, peptide, glycosyl, acid anhydride, carbon-carbon bond, carbon-halogen bond or phosphorus-nitrogen bond. For example, carboxypeptidase. The structure is given in the figure. It is a zinc-containing metalloenzyme which catalyzes the hydrolysis of terminal peptide bond at the carbonyl end of the peptide chain. Phosphatase. It is a zinc, copper, magnesium containing enzyme and act upon organic phosphates to hydrolyze these into free phosphate. Next example, aminopeptidase. It is a magnesium manganese containing enzyme and hydrolyzes polypeptides at the free amino end of the chain to form lower peptides and sometimes free amino acids. Oxidoreductases. First example, oxidases. These are the enzymes those catalyze the removal of hydrogen from a substrate but use only oxygen and hydrogen acceptor. They invariably contain copper and form water as a reaction product. Two important examples of this class are cytochrome oxidase and phenolase. Next example is nitrogenases. The evidence available on bacterial nitrogenases point to the activation of molecular nitrogen by metal ions molybdenum, and non-heme iron and acid labile sulfide groups are thought to be essential for bacterial nitrogen fixation. The nitrogenase of C. pasteurism, A. wendellii exhibit hydrogenase activity towards triply bonded substrate. It appears that molybdenum participate in the action of these enzymes in the form of cofactor. Hydrogenases. Some nicotinimin coenzyme dependent dehydrogenases have been found containing zinc. Superoxide dismutase. During aerobic metabolism, toxic free radical is generated and it is needed to remove this free radical from the system. Cytosolic superoxide dismutase helps in converting superoxide radical into water and oxygen and works as an enzyme. Oxygenases. These are the enzymes that catalyze the direct transfer and incorporation of oxygen into a substrate molecule. Oxygenases do not take part in the reaction. These provide energy to the cell rather than involving in synthesis and degradation of many different metabolites, dehydrogenases or reductases. There are two different types of dehydrogenases known as aerobic and anaerobic. Isomerases and synthesis. These enzymes catalyze interconversion of optical, geometrical or position isomers. Most important example for this category is vitamin B12 coenzyme, whose structure is given in this figure. Summary. Radiant solar energy enters biosphere using magnesium porphyrin complex, that is chlorophyll. The agents responsible for iron transport into and within bacteria are known as siderophores. Important storage and structural metallobiomolecules are ferritin, transferrin and ceruloplasmin. Calcium, silicon and phosphorus play important role in structural metallobiomolecules. The function of oxygen transport and storage in higher animals is provided by iron porphyrin complexes, hemoglobin and myoglobin. Enzymes forms metalloenzymes, ternary enzymes, metal substrate complexes catalyzing different reactions.